We investigated the microinvertebrates that live on the macroalgae in the Great South Bay because we live around there.
Our main idea was to search for extirpated species, since New York is known for their extirpated species.
The biodiversity of ants in the home versus biodiversity of ants like we find outside.
The microbiome analysis of water with varying salinities on the south shore of Long Island.
We collected samples from the Peconic River estuary, and we had all different things. We had dragonfly larvae, snails, leeches, crazy stuff.
We used the queen, the drones, and then the worker bees, and we just compared the different microbiomes of each of them, even though they're from the same hive.
We're basically documenting the succession of how the ecosystem will repair itself after the damage that has been done.
So we did the whole process of like PCR and electrophoresis, and then we sent the samples that were positive to Cold Spring Harbor, and they told us which of our samples should be sequenced. Well, they sequenced them for us, and then we used DNA barcoding to look at them.
DNA barcoding is used to determine the different types of organisms that live within an ecosystem.
This is actually my first science research project.
Since we are freshmen and we all decided to take this elective, this is a whole new world for us, and it's great to be able to be part of this program because you get one foot in the door, whether you want to be a scientist or not.
Last year we did the invertebrates of Gardner Park, so we felt that a continuation and going a little deeper into what's in Gardner Park and trying to find which organisms may be pathogenic in the area. So we decided that a microbiome analysis would definitely be something that could get a lot more specific, and instead of the Sanger sequencing doing invertebrates, we were able to use Illumina sequencing.
When we got our samples back from the Cold Spring Harbor Laboratory, that was really exciting also, because it was the first time that we saw what we had in our hands we now saw on a computer screen with a bit score and with all these different things that go along with that sample, which you would have never even known if you hadn't gone through this whole process.
I am very happy to say that we do have a novel sequence, and we do have two species of the same snail that give birth to live young, and we do have this one snail that miraculously is located on opposite ends of the world.
We didn't find the extra species that we wanted, but finding these bioindicators really went beyond our expectations.
I like that we're able to do our own experiment.
You get to come up with your own curriculum.
There's guidelines set, but really you're the captain of what you're doing here. It's not really set by the school.
For me, I take AP Biology, and this kind of connects with how we do our research, and actually helps me a lot. But for this research, it was more hands-on, and you can get the real actual experience.
In regular labs that you find in school, like in physics or Regents-style classes, you know what that outcome is. But with this, that outcome is a wild card. So it's great how you can formulate your own conclusion as well.
Here we have to actually research ourselves, and we get a deeper knowledge of a smaller topic, but in more detail.
With this project, you actually, and this class, you get to get your hands dirty. You actually get them dirty, and you go in the bay and you do all that.
We're getting the actual microorganisms out of an area, as opposed to just learning the hardcore facts about it.
Well, even just presenting this project here today in front of all these people, it's very good for talking to people, getting used to speaking in front of a crowd.
It's a creative process. So you get to formulate your own project, you get to go through everything on your own, and at the end you can say that, wow, I wrote that, and I'm proud of myself at the end. We are all proud of what we did.
I've loved every minute of it. I don't regret anything.
I think it's great that even though it's our first year, we are able to say that we're experiencing the real science of the class, and it's not, I feel like it's kind of from a distance until you do it.
We actually learned how to do all their programming this year, and the last couple of weeks we've been learning that.
My favorite was probably being in the lab, actually, at Brookhaven National Labs, doing the PCR processes and stuff. It was really educational and really interesting.
I think it's a good start for me to get into research, or I want to do something in science, but I'm not sure what I want to do, so I think it's a good start for me.
I think it could open up more job opportunities for us, because since we have the background with the DNA barcoding and all the programming, we'll be able to better get accustomed to other jobs.
after doing this, I'm interested in the microbiome of a brain, a human brain, so I think it would help me with the knowledge.
This is basically a look at what my future looks like.
Jupiter was definitely difficult.
This is like all pretty and stuff on the poster, but it was really difficult getting everything onto the poster.
This other stuff you got to implement into your writing. So, you know, it was just a little difficult to make it sound as interesting as we think it is, but I feel like we kind of hit the nail on the head in that aspect. By the end of this, we realized how we're supposed to be writing and speaking to people.
Don't let this off to the last second.
Just work at it. It may be like persistent and kind of annoying after a while, but like overall, it's such a great experience to have, and to be able to tell colleges how you've worked with such in-depth like processes, which is amazing.
Try and choose a topic that you actually like. If you're researching something that you think is interesting, it's a lot easier to stay the course.
Try to get a large, diverse array of samples, because the more you get, the larger chance you have of getting a novel sequence.
To not get stressed over it, it's really fun, and when you collect the wet lab data, you should be really careful sometimes, because our data didn't come out right. We had to do it 2 times.
Definitely be patient in like the Jupiter process, because it's very confusing at first, but you'll get used to it and it'll be fun.
People should grab more samples in the future so there will be like less mistakes and just have a more fun experience. You know, it's actually at first when we're at the end of the research, it was being more frustrated after the blast results, and you know, trying to find what species is this and which is that. But all I just recommend is just try to have a great time with the research.